Today we will discuss about the nucleotide and nitrogen base synthesis. The nucleotide and nitrogen bases are synthesized in two ways. First is a de novo synthesis, and second one is salvage pathway. In de novo synthesis, purines and pyrimidines are synthesized from smaller precursor molecules, synthesized from the smaller precursor molecules. And PRP, phosphoribosole pyrophosphate, PRP, which we will discuss in detail, is added to de novo pathway. Added to de novo pathway at some point. Is added at some point of de novo synthesis. While salvage pathway, preformed, already formed purines and pyrimidine bases are are added or converted into nucleotides. Here is here is a question: why we need nucleotides? So the answer is a simple. Nucleotides are needed for DNA synthesis, RNA synthesis, and for energy transfer. Nucleotide energy are used in chemical reaction, otherwise that chemical reaction does not take place. Now I am going to discuss about the de novo synthesis of the pyrimidine bases. So this is basically the super high yield stuff for the USMLE step 1. So what happened in de novo pyridamine synthesis? Carbon dioxide, glutamine, and ATP uh, react with each other uh, to form carboamyl phosphate. This compound is produced and catalyzed by the carboamyl phosphate synthase 2 enzyme. This is a rate limiting step. So first thing, this is a rate limiting step. Second important thing, carboamyl CPS2 and CPS1. So carbamyl phosphate synthase 2 and carbamyl phosphate synthase 1. This is present in a mitochondria and catalyze the reaction in urea cycle. While this CPSP2 catalyze this conversion and present in the cytoplasm. So next step is carbamyl phosphate is convert into the orotic acid. This reaction is catalyzed by an enzyme called dihydro dihydrorotate dehydrogenase. So this enzyme is inhibited by a drug called leflunomide, leflunomide, which is a class of drugs of DMARD. This is DMARD is a disease modifying antirheumatic drugs. So this drugs is used in rheumatoid arthritis. Next, this orotic acid is converted into the UMP, and this reaction is catalyzed by UMP synthase. Again, this is again a super high yield stuff. So deficiency of this UMP synthase occur in orotic acid urea. Orotic acid urea is a autosomal, is a basically the autosomal recessive disorder. So what is deficient in uric acid urea? This UMP synthetase, and this uric acid urea is autosomal recessive disorder. So this UMP is converted into the UDP and then ultimately into this nucleotides are reduced into the deoxy form. So the ribonucleotide reductase is an enzyme which convert these nucleotide into the deoxy form. So this is uridine and this is cytosine. So this uridine diphosphate is converted into uridine monophosphate, and this is ultimately converted into the thymidine. So what does and this UMP conversion into the thymidine is catalyzed by thymidylate synthetase. So thymidylate synthetase is inhibited by a drug called 5-fluorouracil. This is an anti-cancer drug. So another, this to work properly, thymidylate synthetase work properly, they need tetrahydrofolate. So they need tetrahydrofolate. So tetrahydrofolate is synthesized by dihydrofolate, which is converted into the tetrahydrofolate by the enzyme called dihydrofolate reductase, and then ultimately convert into the active form tetrahydrofolate. So this enzyme is inhibited by methotrexate, which is a anti-cancer drugs in eukaryotes like humans, and this enzyme in prokaryotes, dihydrofolate reductase, is inhibited by a drug trimethoprim, and in case of the protozoa, this enzyme dihydrofolate reductase and inhibited by pyrimethamine drug. So this is basically the super high yield stuff for the USMLE point of view. Now I'm going to discuss about the purine bases, de novo synthesis of the purine bases. So basically, this ribose 5-phosphate is comes from HMP shunt, hexose monophosphate shunt. So this is come from HMP shunt, ribose 5-phosphate. Then this ribose 5-phosphate is converted into the phosphoribosyl pyrophosphate by the enzyme PRP synthetase. So again, this is a super high yield stuff. PRP amidinotransferase is a rate limiting enzyme. So again, the substance which inhibit this enzyme PRP amidinotransferase, these are the exogenous substances, are the some drugs, and these are the endogenous substances which inhibit this rate limiting enzyme PRP amidinotransferase. And so these are drugs allopurinol and mercaptopurone. So these drugs, allopurinol drugs, is used in gout. So again, this PRP is converted into 5-phosphoribosyl amine, and then this reaction, this there's a series of reaction converted this phosphoribosylamine into inosine monophosphate and hypoxanthine. So IMP is basically the main product of this reaction. So this is either converted into the GMP or AMP. The conversion of GMP is from conversion IMP is converted into GMP by enzyme called inosine monophosphate dehydrogenase. So this is enzyme which catalyze this reaction, lead to the conversion of the inosine monophosphate into the GMP. So they are the drugs which inhibit this inosine monophosphate dehydrogenase. The drugs are mycophenolate, mycophenolate and ribavirin. Now I am going to discuss about the salvage pathway. So what happened here is basically that this is a nucleic acid which come from the diet and cell turnover. So this nucleic acid is catabolized into the nucleotides and then nucleosides. When the phosphoric acid is removed from the nucleotide, then it is known as a nucleoside. So nucleoside is basically composed of the ribose, sugar, and nitrogen base, and this is a free base. So the what is a salvage? Salvage is the basically synthesis of the nucleotide from the preformed bases. So this is a salvage pathway, the synthesis of the nucleotide from the preformed nitrogen bases. So here is the example when the nucleoside is catabolized into the GMP, and this GMP is further catabolized into the guanosine and guanine. Then this guanine is converted back into the GMP nucleotide by the enzyme called hypoxanthine guanine phosphoribosyl transferase. So this reaction is catalyzed by this enzyme, which is deficient in a condition known as a Lesch-Nyhan syndrome. So again, let so basically we as we already discussed about the de novo synthesis of the purine. So what happened in the purine synthesis? This adenosine is which is come from the AMP and is converted into the inosine. So basically the deamination so of adenosine convert the adenosine into the inosine. Then this is catalyzed by the enzyme adenosine deaminase deaminase enzyme, which is deficient in deaminase deficiency disease, where so which is the one of the major cause of the SCID. SCID is a severe combined immunodeficiency disorder. So this is again ADA deficiency is again a super high yield stuff, and the hypoxanthine guanosine phosphoribosal transferase is again a super high yield. So this anosine is converted into the hypoxanthine and ultimately into the xanthine. So this again, this hypoxanthine is again converted back into the inosine monophosphate. So this is a basically the base. So this base is converted into the nucleoside. So again, what is the salvage pathway? Salvage pathway is a basically formation of the nucleotide from the preformed bases. So hypoxanthine is a base and guanine is base. So conversion of hypoxanthine into the inosine monophosphate is catalyzed by HGPRT enzyme, which is a deficient in the Lesch-Nyhan syndrome. So again, Lesch-Nyhan syndrome is a super high yield stuff. So the this is basically the drug cladribine and pentostatin, which inhibit this enzyme, adenosine deaminase enzyme. So now I am going to discuss about two drugs as well, allopurinol and febuxostat. So basically both are inhibiting of the xanthine oxidase inhibitor. Both are the xanthine oxidase inhibitor. So what is the difference between these two drugs, allopurinol and febuxostat? So allopurinol is basically a purine base inhibitor of xanthine oxidase inhibitor. Allopurinol also inhibit the PRP amidotransferases, which is a rate limiting enzyme in the purine synthesis, de novo purine synthesis, while the febuxostate is only inhibit the xanthine oxidase enzyme. So, in case of the Lesch-Nyhan syndrome, patient present with intellectual disabilities, self-mutilation, aggression, hyperuricemia, and gout. Why gout? Because of the increased production of the uric acid. So this hypoxanthine is not converted back into the IPN. There is increased hypoxanthine which converted into the xanthine and ultimately convert into the uric acid. And uric acid crystal accumulate in the joint. So and in a case of adenosine deaminase deficiency, ADP is required for the degradation of adenosine and deoxyadenosine. So decrease ADL lead to the increase deoxy ATP. So this lead to the decreased ribonucleotide reductase activity and decreased DNA precursor cells and lead to the lymphocytes. So basically, this is the basically the again these are two disorder are the super high yield stuff for the USMLE point of view.